Shall we start? We shall be waiting. Anyway, we shall start with the today's targeted topic, that is culture media. Now, all of you, can you unmute yourself and just tell me what is culture media, whatever you know about it? Can anyone please tell me whatever you know about culture media? Yes. Whatever basic understanding you have. No one? Yes, Surabhi.
Good afternoon, sir.
Good afternoon.
Sir, it is a mixture of nutrients and other substances that help in the growth and survival of microorganisms.
Great. Rather, it is only actually for artificial cultivation. Survival, hardly there are some medium which is used for holding them or making them survive. Okay, anyone else? Anyone else who can define or who can give some more input? Yes, Rutam.
Yes, sir. Good afternoon, sir.
Good afternoon.
Sir, culture media means, like Surabhi said, a medium to cultivate any organisms in the laboratory which is free of any other substances, like any other cells or like that. Pure culture of that bacteria or like that.
True. In other words, you want to say that usually the bacteria or other organisms in nature are present in mixed variety, right? But when you want to get them in pure form or a isolated form where only that same strain exists, okay, at that time you need culture media. Am I understanding right, Rutam?
Yes, sir.
Okay, that is a correct concept. Let me just take you all through the topic which we are planning to have today. Okay, these are the important points which we would like to cover in today's presentation in brief. We have basically the introduction of the media and little bit about the history, what the microbiologists have done till date to employ the artificial culture system to various organisms till date. And we would also see the use of solidification agent agar and how the solidification agent evolved and it has reached to agar, and how the agar we find the best solidifying agent right now. We also will see to the basic constituents of media, and we also will see the types of culture media, right? So let me just introduce you. Bacteria have to be grown or cultured for them to be identified. That is the basic need. Why do we need to grow them in artificial culture medium? Is we need them in little more mass, and number two, we need them in isolated form. What do we mean by isolated? where only that particular bacterial strain or that particular culture exists. Or in other words, it could also be said that organisms or a mass of organism derived from a single parent cell. Okay, that is known as the pure culture, right? Or where we have isolated culture. Now, what happens is, as one of your classmates also mentioned before, and we also discussed, usually if you consider urine specimen, this is just an example, not only urine, any specimen, if you consider human specimen, usually the organisms are present in mixed form. What do we mean by mixed form? There might be more than one bacterial strain which is there. There might be more than one particular bacterial cell is there in a specimen. But what is our interest is we want them to get it in pure and purified and isolated form so that we can study them with various biochemical characteristics and various antimicrobial resistance. Okay, so that is the need. Why do we need to grow them? And we want to have them in a little larger mass, but in a pure or isolated form. That is the objective behind growing this organism. Till date, several type of culture media has been employed to grow them. Originally, the first attempt was done by Louis Pasteur, who employed urine and meat broth as the liquid media to grow the organisms. Liquid media have many disadvantages. What is the drawback of liquid media? Bacteria grow diffusely in liquid media and do not exhibit any specific growth characteristic, and grow in a mixed population. Again, as you add the sample which is having mixture of one, two, or three, or more than that strains of bacteria, different bacteria, then naturally all three or four will be growing together in that particular liquid media. And for us to study them, to get them in an isolated form, it is not possible to use liquid media. That will not help us anyhow. And if you take an example of mixed growth, you can see the first diagram, left side diagram. There are small spot or small medium sized spot which you can see. Now these are nothing but what are they? Can anyone tell me? What are these which you can see on a petri dish of a solid medium? Can anyone? What are these spots means? What does they mean? They are colonies. Okay, they are colonies of bacteria, one particular bacteria. Okay, now what is colony? Can anyone? What is colonies? What is colony? Correct. It is a group of similar bacteria, or rather I would say it is the mass of bacteria which is the outcome of single parent cell, or the place where you find one colony. That means that at that place we have inoculated one particular organism, and that single cell has consumed the nutrition in the petri dish, from the petri dish, and that has given rise to a huge number of progeny cells which has divided from 1 to 2 to 4 to 8, 16, 32 likewise. And that mass of cells which has been derived from a single parent cell, it has become so large that principally that one cell is actually microscopic, but it has grown that much number of bacteria, so as now the colony is macroscopic. The collection of the cell is so huge that we can see them through naked eyes. Okay, so that this is just to compare with a pure growth and mixed growth. The left plate is containing different color, different size, different textured colony. What does it mean? Means individual colonies are derived from a different bacterial cell. So that is example of mixed culture. In the right diagram which you see here, you can see all the colonies are almost similar in color and texture. though they are different, slightly different in size, but that may be group of cells or single cells. That is example of pure culture. Okay, so still liquid media can be used, but we may just employ them to attain the growth of the organism or mass of the organism in very high or very large scale. Whenever we need to have such organisms growing extensively for certain interested compound or medication or vaccine or any other physiological property, whenever we need them to grow in large volume, large mass, at that time we can use liquid media preferably. So on solid media, a discrete colony will be given by a single parent cell, which is originally deriving from a single common parent cell. Okay. On solid media, these colonies may have certain specific or peculiar characteristics like pigment production, hemolysis on blood agar. These are the characteristics which will help us to identify that particular organism, because on certain media, certain bacteria give fixed kind of colony characteristics, and these characteristics will help us to lead towards the organism. The earliest solid medium was cooked cut potato used by Robert Koch. Later he introduced gelatin to solidify, but it was not successful because gelatin used to liquefy at 24 degrees centigrade, and normally we grow the organisms at 37 degrees centigrade, which are of medical importance. Okay, so this is how the requirement of the solidification of the media came and we attempted. Okay, this is just example of solid media. The left diagram which you see, you can see that yellowish orangish color small colonies and colorless medium or white colored medium. Okay, colorless to opaque medium. So this is one characteristics where the organism is producing golden yellow or orange color pigment. In the right diagram where you see there are shining small colonies, and near that colony you can see greenish color pigmentation or discoloration. Rest of the medium which is not been inoculated is red in color because of the sheep blood which is added into it for the enrichment purpose. So that is a blood agar. The diagram of right one photograph which you see is of blood agar, and there the alpha hemolytic organisms are growing in the form of a discrete colony. Okay, so this is just example. Now after employment of various solidification agent, ultimately the discovery of agar powder was breakthrough in the medium solidification. Why do we need to have solidified media? There is one reason. If we get a solidified media, we will be able to grow the organism on some flat surface, and that surface growth will help us to identify them and get them in a huge quantity, plus we can separate them if they are present in mixed culture. We can get individual colonies on the surface and we can just pick up the one which is of our interest, and the other non-interested colony can remain behind and that can be discarded easily. So solid medium was required, and agar is nothing but it is the cell wall extract of one of the algae. Agar is just a cell wall extract of one of the algae, which is a seaweed, and which has been crystallized and solidified and dried in the form of fibers. And those agar powder, if you mix it with other nutrients of the medium, that liquefy them at 98 degrees centigrade. Principally at normal room temperature, they are solid. But if you mix them with distilled water along with the medium constituent, and if you boil them and heat them at 98 degrees centigrade, then it will liquefy. The agar will melt and it will completely mix with the water and medium. And again, when you take them back at 42 degrees centigrade, they suddenly start settling down or solidifying. They start solidification at 42 degrees centigrade, depending on the agar concentration. So that is the key characteristic of this agar-agar powder, which we are utilizing here in preparing medium. I know many of you may be loving the jam, okay, fruit jam, and applying it on the bread or any other surfaces which is edible and consume them along with your food. Okay, I hope many of you may be knowing, but have you ever asked yourself that the jam bottle is not always a flat one or a kind of straight bottle? They are usually curved bottle, and whenever you pull out the jam from the bottle, it is actually jellified or solidified. And how they would have settled down the jam in those curved bottle, have you asked ever yourself? Anyone? Can anyone tell me, have you ever tried to ask yourself how the jam gets solidified in that curved bottle, and when you get it, it is exactly in the shape of that bottle inner surface? That is nothing but it is because of agar-agar powder. That fruit jam is also containing agar-agar powder that is mixed with the syrup. The fruit syrup is mixed with agar-agar powder. It is then boiled, and after boiling, they simply pour that liquefied content into glass jar, where after cooling down below 42 degrees centigrade, they will settle down and they will take the shape of your jam bottle. So this is how we mix the bacterial medium constituent with the distilled water and the agar-agar powder, and then we pour them in the liquid state only. After boiling them in the liquid state only, we pour them into the medium. And after pouring into the medium, we simply, so pouring into the petri dish, we simply let them cool down. And once they are cooled down, they will be taking the shape of petri dish, and they would have a clear solid media surface which can be used for inoculation of the bacterium. So this is the diagram of agar-agar powder, how it is. So let me just tell you a brief view about basic constituents of the medium. What are the basic constituents of the medium? Principal constituent for any bacteria to grow in a basic medium is source of carbon, source of nitrogen, and salt. Apart from agar, these are the only important source for the organism to grow and for the medium to be defined as artificial culture medium. Now, for the source of carbon, principally, we rely on various polysaccharides, okay, or monosaccharides or disaccharides, okay. For source of nitrogen, we rely on peptone. Now, peptone is nothing but it is the digested, partially digested product of complex protein. So they might have single amino acid or few amino acid chain proteins, okay, and bacteria will consume this peptone or proteinaceous product as source of nitrogen. Apart from this, we add various inorganic salts like phosphate, potassium, magnesium, and certain other growth factors. Okay, so to compositely provide this various inorganic salt and few peptone, we use meat extract, sometimes blood, sheep blood, sometimes sheep or human serum, or yeast extract as a composite crude partially defined medium ingredient. Okay, so let me just tell you about types of media. I'll just hold here. Now, can anyone tell me if you are not clear with anything and if I need to repeat? If anybody has got any kind of query till whatever we have discussed, and if you feel I should be repeating one particular portion of the explained presentation, then please tell me, otherwise we will start from here. If anybody wish to repeat just within next 10 seconds, please tell me, or else I will start. Okay, I'll start then. No problem. Now, based on the consistency, the medium are divided in three varieties: solid, semi-solid, and liquid medium. Liquid medium is also known as broth. Okay. Now, the solid media, semi-solid media, and liquid media. Let me tell you the only difference between solid and semi-solid, and liquid is the concentration of agar-agar powder. Because agar-agar powder will lead to hardening or solidification. In liquid media, we do not add any kind of agar-agar powder because we need them in broth format, liquid format only. So we never intended to have any solidification. So we never add any agar-agar powder in liquid media. On the other extreme, in solid media, we usually add 1.5 to 2% concentration of agar-agar powder. 1.5 to 2% agar-agar in liquid media. If you add, then that media will become solid. Okay. Now instead, if I add 0.75 to 1% of the agar-agar powder, then it will not be completely solid like a solid medium, and neither it will be a liquid medium. So that will give us the semi-solid medium. Okay. So that is the types of the media based on the consistency. So these are three types. The left one, agar-agar plate, which you see is the example of solid media. Then the second one in the test tube, which you see is agar deep tube. Okay, that is a semi-solid medium. The second tube which you see is broth, that is not containing any kind of agar-agar. It is completely a liquid medium. And the third one is agar-agar slant, which is again a solid medium, but simply it is prepared in a test tube format. That is the only difference. Okay. Now let me just tell you about the variety of medium based on its composition. The medium has been divided based on its composition in varieties of type. I will just tell you the number one reason to differentiate them is the its composition, and number two is its use or application for identifying or growing the medium. Now, how it is different, I will tell you with each type. Okay. So simple media are the media which is usually containing one or the other kind of carbon source. The example is nutrient broth. It is consisting of peptone as a source of nitrogen, meat extract as a source of carbon and few salts, NaCl, and water. Adding 2% agar, it gives nutrient agar. Okay, and it supports the growth of all non-fastidious bacteria and used widely. Now, what do we understand by non-fastidious bacteria? What do we understand by fastidious bacteria, and what do we understand by non-fastidious bacteria? Now, fastidious bacteria are choosy bacteria. They are stringent bacteria. If we try to grow them artificially, other compound or condition to consume the nutrition and grow. If we do not provide that condition or that nutrient, that organism will not grow. So that is fastidious bacteria. Non-fastidious are the one which can grow in every basic media. Okay, they do not have any additional or extra nutrient requirement. Okay, so they are relatively simple to grow, non-fastidious. And this simple media grows well the fastidious media. The second one is complex media. These have certain added constituents to bring certain characteristics to provide special growth nutrient to bacteria. Now we have got varieties of complex media based on its application. I will just lead you to that. Okay. synthetic or defined medium. Synthetic or defined medium is again something kind of complex, where each and every constituent of the medium is very well calculated and it is artificially prepared, and then they are mixed together to bring the exact amount of required substance in the medium. So they are a kind of artificial medium where the components or the constituents are completely predefined and they are prepared in that manner only. Enriched medium. Now, what is enriched medium? Now I'll tell you there are two different terminology: enriched medium and enrichment medium. Both are different. Let me just tell you one by one, but both are different, and try to understand the difference between these two: enrichment medium and enriched medium. Let me just tell you what is enriched medium. Enriched medium is a medium which is basic medium, and in that basic medium we add certain extensively nutritionally rich substance for the bacteria or sheep or human serum, or even egg or egg protein. They are added additionally into the basic medium, and then it becomes enriched medium. Okay, because blood, serum and egg are very rich in varieties of sterols, varieties of peptones, varieties of carbohydrates and varieties of micronutrients. So they will support the growth of certain fastidious bacteria which will usually grow only in nutritionally rich condition. So there is a crude enrichment here. You can understand these enrichment will support the growth of almost all the bacteria in a luxurious manner. All the bacteria will be growing in a luxurious manner. Now, example of this enriched medium is blood agar and chocolate agar and alkaline peptone water. Now, chocolate agar, you all may be amazed that bacteria also like to eat chocolate. No, it is the same medium which is known as blood agar. Blood agar is simply prepared by adding 5% quantity of sheep blood in nutrient agar and then mixing well and pouring them. That is blood agar. But before pouring them, after mixing 5% blood, if you hemolyze the blood, if you keep that flask at 55 degrees centigrade for 60 minutes, then all the RBC which is there in the blood will be lysed. So the blood will be hemolyzed and that will be converted into chocolate brown or coffee color. Okay, so that is why it is known as chocolate agar, but it is not having any content of chocolate. Mind well, the chocolate agar is heated blood agar, and blood agar is a simple blood agar. So that is enriched medium. Let me tell you about enrichment medium. Now, what is enrichment? Enrichment medium is also having some very well-defined single constituents in extensive amount in the basic medium, not the whole enrichment like addition of the blood, serum or egg is done. We only add or increase the concentration of one particular content of the medium. What it will do is, I'm telling you with an example. Say, for example, if we have an opportunity of cultivating bacteria from stool of any human. Say, for example, we are going for stool culture. Usually in human intestine, there are a lot of varieties of commensal or normal flora organisms survives. Okay, the best example is, anyone knows normal flora of human intestine? Anyone? Normal flora of human intestine is E. coli. Yes, it is E. coli. E. coli, which is seen commonly as a normal flora of human intestine. But whenever the patient is infected with, say, for example, Salmonella, Vibrio cholerae, Shigella, kind of GI tract pathogen, at that time, on culturing the stool sample, what will happen is these Salmonella, Shigella, or Vibrio cholerae, if they are present as pathogen, they will grow on the medium. They will grow. I would say it will not be like they will not grow. They will grow. But the number of commensal or normal flora is extensively high, at least more than 1,000 times higher than the content, than the concentration or amount of Vibrio cholerae, say for example. The normal flora is 1,000 times more in number compared to Vibrio cholerae. So what will happen in the medium? Vibrio cholerae will not be able to grow well, and most of the medium will be covered or it would be consumed by the normal flora E. coli. So you will miss the Vibrio cholerae single colony even if it is there on the meter. Is we want to let the Vibrio cholerae grow normally, but we want to discourage the E. coli to overgrow. We discourage the commensal or normal flora to overgrow and take the nutrition of Vibrio cholerae. So we add alkaline pH into the medium because we know Vibrio cholerae grows very well in alkaline pH compared to the neutral one. But E. coli need near neutral 7.2 pH. In alkaline pH, Escherichia coli, the normal flora, will not grow. So this keeping the alkaline pH will let the Vibrio cholerae grow normally or sometimes very well luxuriantly, but it will not give the interest to commensal. So this is how the enrichment medium works. This will selectively favor the organism of our interest for the growth of commensal. So that is the enrichment medium. Everyone understood well? Yes or no? You can unmute. Yes or no? Okay, that is great. So I'll go back. Enriched medium, where the medium is luxuriantly nutritious because of certain substance like blood, serum, or egg. Okay. Enrichment medium is the one where selective support to the organism of interest is provided. Okay. And the commensals are relatively discouraged. The example is tetrathionate broth for Salmonella, selenite F broth for Shigella dysentery. Okay. Also the alkaline peptone water. So this is just a image of selenite F medium. What is selective medium? Selective medium is a medium which will let only and only the organism of our interest to grow, inhibit the organism which is not of our interest. Similarly, we take the same example of stool culture, an example of deoxycholate citrate agar. The deoxycholate citrate agar will support but they will not let E. coli to grow even a single colony. So there the advantage to the organism of interest is very high, and we shall be able to grow the organism of interest like Vibrio cholerae, Salmonella, Shigella in a pure form, even if they are very minute in number. Okay, so that is selective medium. Indicator medium. Indicator medium is the medium where with the growth of certain organisms, it will give one particular color to that particular colony. Say, for example, if I take the example of Wilson and Blair medium, on that medium, if the colony of Salmonella grows, it will give black metallic sheen to that particular colony. And if other colonies of E. coli or Shigella is growing, it will not be having black metallic sheen. So easily the organism of interest can be indicated. Okay, we will be knowing that the organism of interest has grown. Okay, similarly for Corynebacterium blood agar, okay, potassium telluride agar, if the Corynebacter diphtheria bacillus grows, it will give you the black jet colored colony, and rest other organisms will not be giving black jet colored colony. So the left one, the image is very hazy, but it is the example of Salmonella shigella agar. And the right one image which you can see here is the example of blood tellurite agar, where the colonies of Corynebacter diphtheria, which is there in the form of black color. Now differential medium. What is differential medium? Differential medium do not inhibit or do not discourage any of these organisms. They will not let all the organisms grow properly. But based on its digestion capacity, its biochemical property, different group of organism in two varieties, okay, with some color or something like that. The example here is McConkeys agar. Okay, in McConkeys agar, all the gram-negative bacteria will grow very well. Okay, but in gram-negative bacteria, we have got two broad groups. One is lactose fermenting, another one is lactose non-fermenting. Okay, the lactose fermenting gram-negative bacteria will give you phenol red. Okay, because if the lactose will be fermented, it would produce lot of amount of acid, and the acid will bring the pH nearby the colony towards acidic, and the phenol red indicator will give the pink color. Okay, but if the organism is lactose non-fermenting, then it will give pale or colorless colony. So this is the difference between lactose fermenter and non-fermenter, achieved with the help of pH indicate McConkeys agar, and that's how the McConkeys agar is known as differential medium. Similarly, on blood agar, we can have three varieties of colony. One is alpha hemolytic, second one is beta hemolytic, and the third one is non-hemolytic or gamma hemolytic. Alpha hemolytic colonies will be greenish, as according to one of the image which we saw before. Okay, alpha hemolysis is a kind of partial hemolysis. So the organism which is only capable to break down alpha chain of hemoglobin, they will give greenish colored zone surrounding the colony. So that is alpha hemolytic colony. Beta hemolysis means complete hemolysis. Beta hemolytic colony will be having colorless zone. Complete RBCs would be lysed. So colorless zone surrounding that particular colony. And the gamma hemolysis means no hemolysis. Okay, in that the organism in that colony will not be altered at all. It won't be colorless, or it won't be even greenish. It will be as it is, red in color. So that blood agar is also one of the type of differential medium, where it will help us to differentiate alpha hemolytic, beta hemolytic, and gamma hemolytic organisms. Sugar media. This is simply the medium used for detecting the acid and gas production from 1% sugar, that is digest or ferment that particular sugar or not. So this is simply sugar media. Transport medium. All of you may have nowadays, I mean, heard and knowing well what is viral transport medium. Okay, for various samples used for RT-PCR or for virus culture is employing viral transport medium. Apart from this, we also have got Stewart's medium, Ames medium. Okay, Stewart's medium is containing non-nutrient gel with reducing agent to transport the sample containing gonococci. Okay, buffered glycerol saline is also used for transporting the stool specimen which is suspected for enteric bacillus like Salmonella or Shigella. Transport medium are the medium which will not let the organism to overgrow. Also, it will not let the organism to die even when you need to transport the specimen from one lab to another lab, and the processing is expected to be delayed. Whenever the processing is getting delayed, at that time you can go for using transport medium. Okay, so that is transport medium. This is just holding medium. This will not let the bacteria to die or not let it hold it. Anaerobic medium. Anaerobic medium is the medium which will support the growth of anaerobic organisms. Okay, example is Robertson cooked meat broth, RCMB. That will lead the anaerobic organisms to grow by creating continuous anaerobic environment because of minced particle of cooked meat. Okay, small pieces of cooked meat is added into the medium, which will keep the medium completely reduced and it will consume all the oxygen. So this is example of transport medium, the left one which you see as well as the right one which you see. So that's it for the culture media which I intended to deliver today and wanted to discuss with you. If there is any query with anyone, we can discuss it at the same moment. Please, if anyone is having query, just let me know.